So by now we are already aware that bacteria becomes resistant. But how do they acquire such ability? In the preceding slides, we will be learning about the mechanism of antibiotic resistance. But before that, let us have an overview of the mode of action of antibiotics. We have to know that antibiotics can either destroy or decrease the growth rate of bacteria. So therefore, if it destroys the bacteria, it is bactericidal, and if it slows down their growth rate, it is bacteriostatic. Here we can see a typical bacteria. Within the cytoplasm, we have circular DNA, RNA being synthesized, and ribosome synthesizing protein. The first class of antibiotic are the ones that targets the cell membrane. Antibiotics like polymyxin disrupt the cell wall function, making it more permeable, causing leakage and thus disrupting the osmotic balance, which eventually leads to cell death. Other antibiotic class targets the cell wall. Drugs like penicillin, cephalosporin, pacitrazine, which have beta-lactam rings, about which I'll be discussing later, uh, inhibits the cell wall synthesis and are bactericidal. Different drugs will have different mechanism, and one thing we should know about this class of drug is it will not affect human cell as we like cell wall. The next class is the one that can inhibit DNA and RNA synthesis. Drugs like quinoline, nalidixic acid inhibits the synthesis of DNA, and drugs like rifamycin blocks RNA synthesis and therefore affecting the protein synthesis. Now coming to protein synthesis, there are other class of antibiotic that inhibit protein itself. We all know that ribosome synthesize protein, and there are certain drugs that target either 30S subunit or 50S subunits of ribosomes. Drugs like tetracycline, streptomycin targets the 30S subunit of ribosome, and drugs like erythromycin, chloramphenicol targets the 50S subunit of ribosome. The last class of antibiotic are the ones that inhibits folate metabolism. Folate or folic acid is essential for the synthesis of adenine and thymidine. Drugs like sulfonamide, trimethoprim prevents conversion of folate precursor, that is BAPA or BABA to folate. One thing we must know is humans do not synthesize folic acid, so these antibiotics are okay to take and are selective to bacteria only. So before I proceed, I would want each one of you to ponder on these questions, and by the end of this presentation, have an idea about it. So, what causes antibiotic resistance? Why does antibiotic become less effective? To answer this, let us have two approaches. The first being the molecular approach. Overuse of drugs can cause random or spontaneous mutation in the central dogma, which will synthesize new protein. This new protein will have morphological or functional change, which would be passed on to the next generation. The second approach is the evolutionary approach. In this, there is an evolutionary pressure to adapt because nature does not allow eradicating all the species, so it must become resistant. The fittest surviving bacteria would undergo natural selection and reproduce to produce new resistant generation. Now, moving on to the next question, how does bacteria generate antibiotic resistant response? Apart from circular DNA, bacteria have an additional genetic material called the plasmids, which usually carries resistant gene. This plasmids incorporates to main DNA, which synthesize mRNA that is read by ribosomes to create new resistant protein. The resistant gene on the plasmid will decide what type of resistance will the bacteria acquire. It can be antibiotic degrading enzyme, uh, for example, beta lactamase. Beta lactamase is an enzyme which is normally found in E. coli. It breaks down the beta lactam ring found in penicillin, leading to inactivation of the drugs. Antibiotic resistance may also produce an efflux pump. An example of efflux pump is seen in antibiotic tetracycline. So, tetracycline inhibits protein synthesis, and this bacteria uses efflux pump to pump out the drug. Some other bacteria also have resistant gene to modify its membrane structure and its binding target. It changes the structure of the protein. A great example of this is seen in MRSA, which stands for methicillin resistant Staphylococcus aureus. Methicillin does not work because the protein that methicillin binds onto is modified within the bacteria. MRSA is a very special strain of bacteria. What makes it special is that it is a superbug that cannot be killed by a number of common antibiotics. MRSA infects different parts of the body, including skin, and in rare cases, it infects our joints and lungs. The range of this infection can vary from limited to serious. When we talk of MRSA, we traditionally associated it to hospitalization or healthcare associated risk. Because of the resistant property they have, there have been many cases of MRSA outbreak uh, recorded in hospitals. People who never had a history of staph infection were infected with MRSA in their later life, and it was later known to all that MRSA was spread through community associated MRSA. It is very easy for a person to contract infection as it spread through crowd, direct contact, or even shared items. Now proceeding to the next question. How do bacteria acquire resistant gene? Well, there are two ways for bacteria to acquire resistant gene. First is the vertical gene transfer. In this, bacteria acquire gene through reproduction. Vertical transmission is spontaneous and it takes time to pass on. The second form of transmission is the horizontal transmission. Bacteria can form pilus and can attach to other bacteria and pass on their plasmids. And such kind of transmission is called conjugation. And conjugation. Another form of horizontal gene transfer is the virus transfer, or what we normally known as bacteriophage transfer. So what happens is bacteriophage injects its phage DNA into the bacteria, which and incorporates itself to bacterial DNA. It destroys all the bacterial DNA, leaving the resistant gene floating, and it replicates its viral DNA into the bacteria. Later, it incorporates the viral DNA and lyses the bacteria and releases itself into the environment. So, what's special about this newly formed phage is it consists resistant gene which are passed on to other bacteria as the bacteriophage continues its cycle. Last type of transformation, last type of horizontal transmission is transformation. When resistant bacteria die, bacteria picks up the genetic material from the environment and incorporates a resistant gene into itself. So these are the important ways in which bacteria acquire resistant DNA. Thank you.